Welcome. No worries. All right. Well, what a wonderful group of people. Thank you all for coming. It's my great pleasure to MC this session. My name is Hans-Peter Pfister. I'm Jakob's PhD advisor. And we have our committee members here, Professor Paul Zickler and Professor Elena Glassman. So the way this works is Jakob will give a presentation for roughly 45 minutes. Anyone is free to interrupt with any clarifying questions at any time. And then afterwards, we have time for some audience questions. And then we'll ask you to leave the room, don't go too far, and the committee will grill Jakob for another 2 hours. And then after our questioning, we'll convene the committee. Jakob will come out first, so don't get too excited. The committee still has to deliberate, and then we'll come out and announce the decision. So with that, I'll hand it over to you.
Thank you, Hans-Peter. Quick check on Zoom. People can hear me all right? Can I get a thumbs up from someone? Okay, great, fantastic. It's amazing to see so many familiar faces. It's been so much, so great to have you all here. I am excited to tell you about my PhD research that is on building computational and scalable computational tools for connectomics. Now, before I get started, I want to, overall motivate my research. And in biology, in biological research, a key goal is to relate the structure of an organ or the anatomy with its function. So, for example, if you take a look at the brain, at the heart, we can see by looking at its anatomy, at its muscles, we can see what its function is supposed to be. So the mechanistic insight we're getting is that the heart is pumping blood through our body. And now the only organ in our body where we do not have this sort of structure to function mechanistic understanding is the brain. And the reason for that is that with the brain, we don't really know actually how the anatomy at a neuron level actually looks like. So in order to address that problem, in connectomics, we build and analyze very, very detailed maps of brains or entire nervous systems even. Now, the largest connectome that we have today that covers an entire body or an entire central nervous system is the brain of a fruit fly. And so what you're seeing in this video is a rendering of that connectome with the brain over here, and this bulb over there is the ventral nerve cord that's somewhat similar to the spinal cord of humans, and they're connected through this neck over here. And now every color or every object in this map is one single neuron, and for each of those neurons, we know how they're connected with each other. And so to orient you a little bit, this is actually a pretty cool angle. So this is the brain of the fly, and these bulbs over here are called the optic lobes. So these are the neurons that process visual information on the fly. So if you squint your eyes a little bit, you can actually, see the fly looking at you here. And now, how do these maps give us a more mechanistic understanding of how the brain works? So here's a simple example of how we can now use that connectome to trace a pathway that takes in optical input through the eyes of the fly, and then how that information is processed all the way to the body of the fly, where we can then, or where the fly then uses that input to respond with certain motor control, for example, like wing movement or moving its legs. Okay, so connectomics has gotten lots of attention in the past years, so much that the scientific journal Nature has dedicated 2 special issues just for reconstructing and analyzing connectomes. The first one was last year that was entirely dedicated to the fruit fly connectome, and a second special issue in Nature that was entirely dedicated to studying cortical tissues in mice. Now, the largest connectome of an entire body is that of a fruit fly. But what we can do today is we can look at small portions or small subvolumes of human brains. And I was lucky to be part of a research study that was published in Science magazine last year that reconstructed a cubic millimeter of human temporal cortex brain tissue at nanoscale resolution. That project was led by my colleagues, Jeff Lichtman and Viren Jain. And today, this reconstruction is the most detailed human brain reconstruction today. So, and that's why it also received a lot of media attention. For example, it received a New York Times Good Tech Award. It was covered by The Guardian, CNN, and also shout out to my parents for spotting it in the German news. Okay, so how do we actually reconstruct or get a connectome? Typically, we would start by extracting a small piece of brain tissue, either from a human or an entire brain from a fly or a mouse even. And then that piece of brain is then sliced into very, very thin layers. These layers are then imaged usually with electron microscopy, or more recently also with optical microscopy types of approaches. Then these images of each of these brain slice layers are aligned and stacked on top of each other to form a three-dimensional digital volume. Then that volume is segmented. So for each voxel in that volume, we assign a label, and that is typically done using 3D convolutional neural networks. And once the AI has done its segmentation job, we still today need to perform something we call manual human proofreading. So we need to correct certain reconstruction errors that the AI has made. I will talk a lot about this in this talk today. And then once we are certain that the reconstruction quality is at a certain level, we can then actually look and analyze the circuit architecture of that connectome. And maybe another way of thinking about this pipeline is actually that each subsequent step in that pipeline introduces another level of data abstraction. So for example, when you image a slice of brain, you discretize it, you put it on a bitmap in a pixel, and then we keep abstracting it all the way until in the end we have a graph or a network that describes that piece of brain. So to get from all the way on the left to the pipeline to all the way on the right, currently today is a very labor-intensive process. So for the frontier data sets, this can take up to 10 or more than 10 years of work to reconstruct a large-scale connectome. And in my work, really my goal is to enhance the computational steps in that pipeline so that hopefully in the future we can go through that pipeline in a matter of weeks rather than tens of years. Okay, so in order to achieve that goal, we first need to understand what are the current computational bottlenecks in connectomics. And I want to outline three of the most important ones today. So the first bottleneck is called the data problem. So connectomes are really large in terms of their data size. And I think it's fair to say that the size of connectomics data set in terms of their memory requirements have grown exponentially over the past year. So for example, the fly over the past decade, they've grown exponentially. So for example, the fly brain roughly covers around 100 terabytes versus the H1 data set, this is the human cubic millimeter that I mentioned earlier, is over 1.4 petabytes in memory size. And the next big goal in the connectomics community is reconstructing a mouse brain connectome. And that would lead to around 1 exabyte of data. And I intentionally didn't put a date here yet because it is yet to be achieved. So it's unclear when we will get that connectome. Okay, so that's the data problem. The second problem that I want to highlight is called the proofreading problem. And that is that manual error correction in neuron reconstruction is an extremely slow process. So after segmenting a data set, we would, for example, get a reconstruction like this. And a skilled proofreader or a neuroscientist can very quickly tell that this is not actually one neuron, but 2 neurons. And the way we currently have to correct for these errors is for a human to manually go in and then manually correct these reconstruction errors and split those 2 neurons apart. And finally, the third problem that I want to highlight is the analysis problem. And that is, in simple terms, connectomes are very complex. So even if you have a fully accurately reconstructed connectome, it is very challenging to sort of extract meaningful insight just from looking at the circuit architecture, and that also requires work. Okay, so over the course of my PhD, I've tackled these bottlenecks mostly using tools from applied machine learning and interactive data visualization applied to this field in neuroscience. And I've made several contributions, some that are primarily predominantly contributing to machine learning, some that have primary contributions in interactive data visualization, and also larger scale group projects that make primary contributions in neuroscience. Now, for this talk today, I want to go over the main contributions of my thesis. And the first contribution that I'm making in my thesis is addressing the data problem. So I've been developing machine learning-based algorithm for isotropic volume reconstruction. That is, how can you get higher resolution data just from lower resolution microscopy input? The second contribution is addressing the proofreading problem, where we are proposing a algorithm that can reason over entire neuron shapes in order to detect and correct certain reconstruction errors. Then the third contribution is an interactive tool for network motif analysis in connectomes. And the final contribution is another interactive tool that studies motif, but combined with morphology, like neuron morphology and network connectivity. Now, for the sake of time today, I want to, okay, so here the first two contributions are learning-based algorithms, and the second contributions are in the interactive data visualization based. So for the sake of time today, I want to focus on my contributions that relate to the proofreading problem and the analysis problem. And I will start with my neuron shape reasoning-based approach that tackles the proofreading problem. Okay, so in order to better understand how we can tackle this, we need to first see what types of reconstruction errors does an AI-based neuron segmentation algorithm make. And the first reconstruction error is called a split error. So in this small example, you can see that the segmentation algorithm has incorrectly segmented this one particular neuron as two separate segments, but instead, what it should be is one segment. So these two segments need to be merged together. And the second type of error is called a merge error. And for this type of error, we, in order to correct it, we need to split those two segments apart such that they accurately reflect the neural anatomy. Now, in 2D, those errors, you may ask, like, oh, why would I care? Maybe it doesn't really make such a big of a difference if I just leave these reconstruction errors in the data set. But if you look at those errors in 3D, you can see that actually they're making like quite a significant change to the neural circuitry in the connectome. So for example, if I were to like just leave off this green branch over here, I would miss out all the synaptic input or outputs that this branch is making to other cells. So it would just not be as good of a data source. And similarly for merge errors, here you can see that this neuron has been incorrectly merged with another cell. And also this would alter the synaptic wiring diagram quite a bit if we leave those uncorrected. So I've been collaborating with folks at the Allen Institute and Princeton on a system called CAVE that we published in Nature Methods this year. And the first type of approach that we took was, can we tackle this reconstruction process interactively? So this is a system where users can go in and interactively correct such a reconstruction error. And here's a screen capture of a user using that system. So they've identified a merge error, and now by marking each neuron individually, they can now split those two neurons apart. So this is a web-based interactive system that stores, or you can think of it sort of as a Git version control for connectomes. So everybody in the world can sign up and can make edits to a connectome, and then those edits are reflected globally for everyone who uses that system. So that's been fairly optimized already. And we've deployed the system at the Lichtman lab here at Harvard on 2 large-scale connectome datasets. One is a fish dataset and one is a human dataset. We received over 250, or we have over 250 users on that system that use it regularly. And collectively, those users, together with some agentic approaches, have made over 90,000 edits to those 2 datasets. So that was a success, but it soon became very clear that just using these interactive or manual approaches, that is not enough to go through these entire datasets. You would have to spend way too much time and money on that. And that is illustrated by this report that came out in 2023 by the Wellcome Trust. That's a funding agency that funds connectomics research in the UK. And they've estimated sort of the cost that is associated with mapping a full mouse brain connectome with the current state-of-the-art technology. And what they've identified is that the proofreading is one of the largest, most driving cost factors of getting that connectome. So they've estimated it to be $7 to $21 billion just to proofread an entire mouse brain connectome, which in order to actually make that happen, we will need to bring the cost massively down for that project, for that sort of like bottleneck in the pipeline. Now, so why are AI-based reconstruction errors still making so many mistakes that we then have to manually fix afterwards? And I think a key contribution to that is that right now, these algorithms are typically 3D convolutional neural networks. And these models have very limited receptive fields that don't really map well to the sparse thin architecture of neurons. So for example, here, so this is one neuron, a fly neuron. And what you can see here is the field of view that a particular segmentation algorithm that's the working horse in connectomics has. And that's very small in relation to how big this neuron is. So if you if you think this sort of algorithm has to step along this whole neuron the whole time, and it's very easy for it to make errors in that process. Like it may miss a branch or something that we then later have to correct. And that's really a trend over all reconstruction methods in connectomics. And what we've been thinking is, could we build an algorithm that essentially can reason over the entire shapes of neurons and sort of learn a shape distribution, and then use that knowledge to sort of tell us, oh, this doesn't really look like a neuron. Maybe you need to make an edit, or maybe the model can even make these edits themselves. And that sort of project is available as a preprint we put online this year. Okay, so how does this algorithm actually work? So we would start off by the output of a 3D CNN, a segmentation volume, and that segmentation volume may potentially still contain quite a few reconstruction errors. Then in the first step, we would sample a point cloud of multiple neuron segments for which we have an estimate of that they most likely may belong together, but maybe also not. We're not really sure. So you can think of this as some sort of over-complete point cloud that may contain multiple neurons, but also fragments of incomplete neurons. And our algorithm then essentially takes this point cloud and relabels the points in sort of a reclustering, such that each of those clusters reflects an entire neuron morphology. And it does so in a learned way. So under the learning objectives of this algorithm are the following. So we start by a multi-neuron point cloud. Then our model called the point affinity transformer takes this point cloud as an input and uses it as context. And for each pair of points, it would then we can then ask the model, how likely is it that those 2 points belong to the same neuron, yes or no? So 1 would mean that 2 points belong to the same neuron, 0 would mean they do not belong to the same neuron. And then if we do that over all different pairs of points, what we're getting is something that we call a point affinity matrix. So each pixel in that matrix tells us how likely it is that 2 points belong to the same neuron. And you can think of this matrix as some sort of point proximity metric. And then if we use that metric to cluster the point cloud, we can then derive new cluster labels and sort of like carve this point cloud into what then eventually becomes individual neurons. Okay, so how do we train this algorithm? I've told you earlier that we deployed this interactive proofreading system where humans edit connectomes. So from these edits, we have a lot of ground truth data from humans that we can use to train our models. And we ask our model to predict these affinity matrices. And then based on the human generated ground truth, we have the ground truth affinity matrix. So it's a supervised approach. And then we can compute the binary cross entropy loss between the predicted affinity matrix and the ground truth affinity matrix, compute a gradient from that, and then backpropagate that through our model. Now, briefly looking under the hood of how this point affinity transformer looks like from a neural network architecture point of perspective. So the model consists of an encoder that takes in point embeddings. And these point embeddings are then fed through a series of cross and self-attention layers that produce a feature set that describe this point cloud. And then in the decoder, we input different point pairs. And based on those point pairs, we can decode a set of affinity scores using this decoder MLP here in the back. And that then ultimately produces this matrix that we then can cluster. Okay, so here are some results of our method. We compared it against like some this is a pretty naive baseline, like just clustering the point cloud using Euclidean distances, which doesn't yield such good results. So we turned then to more learning-based approaches, such as a graph neural network approach and another transformer-based approach. And overall, compared to the ground truth, we found that our method sort of like was able to separate out these multi-neuron point clouds much better. It also works for pretty difficult examples. So here, this is a point cloud that contains 8 neurons, and they all have been like mushed and merged together. And if we apply our model to it, we can relatively confidently carve out these individual neurons from here, which was very encouraging. Here's some more quantitative results on the fly-brain connectome dataset. So we report the results over 4 metrics. And here, every metric, the lower it is, the better the algorithm performs. So compared to Euclidean clustering, the GNN-based approach, a point transformer-based, 2 point transformer-based approaches, and then our approach here in orange, our method outperformed these baselines over all these 4 different metrics. Okay, so summarizing the contributions of this particular project to the proofreading problem. So overall, we presented a neuron reconstruction approach that really goes beyond the sort of conventional state-of-the-art 3D CNN-based image segmentation approaches. We proposed a new neuron shape learning framework that is centered around this pairwise affinity prediction task. And then we applied this shape learning framework in combination with an agglomerative clustering approach to correct neuron reconstruction errors. All right, so that's it about the first part, or tackling the proofreading problem. And now I want to step back for a second and remind you about the Connectomics pipeline. So after proofreading a connectome, once we've, are certain that the data is of a certain level of quality, then we can actually turn towards what we really want to do and look at like how is the circuit architecture, like what are the like motifs or structures of interest in there. And the two parts of the next two projects that I will present are now talking about the analysis problem. So how can we extract certain biological insight from these already proof-read connectome data sets? All right. So the question the guiding question is, how do we actually analyze a connectome? And it turns out that looking at circuit motifs is a really powerful way of studying connectomes. And what do I mean by a circuit motif? So for example, in this network or this example network over here, we see that this example set of neurons really, that there's this like motif in here where 3 neurons form like a circular connection. And that's what we call a motif in connectomics. And if we sort of think of a connectome as a graph where each neuron is a node and the synaptic connections between those neurons are edges, we can apply traditional methods from graph analysis like subgraph isomorphism searches to identify these motifs in a connectome graph. And looking at these sorts of motifs is extremely interesting. So you can think of these small circuit patterns as some sort of atomic units of computation in the brain. And they have been previously mapped to certain behavioral aspects that animals can do. So for example, some of my colleagues at Janelia Research Campus have studied the central brain of the fruit fly and then identified certain network motifs that allow the fly to encode direction in space and also flexibly navigate. Okay, so they're very interesting. We know that to a lot of people, and nonetheless, they're very hard to study. And a reason for that is that the connectome data sets are really large. They can have tens of thousands of nodes. They are very dense with over 130 million edges. And performing these subgraph isomorphism searches in those connectomes is an expensive computation to perform. So for example, verifying whether a certain motif exists in a graph is an NP-complete problem. And finally, the complex three-dimensional structure of the nodes in the network, the neurons themselves, have very complex branching patterns. And I think that makes this problem especially interesting from a data visualization point of perspective, is that the graph itself inherently has some sort of spatial dimension to it. Okay, so based on these requirements, we've built an interactive analysis tool called Vimo. And in Vimo, users can sketch these like small circuit motifs here on the top left, and then look at instances of that pattern, how they're implemented in a connectome. So in this case, this is an example from central brain of the fruit fly with an external ring neuron that the fly uses to also encode direction in space. So in this project, we designed Vimo in collaboration with lots of neuroscience domain experts, and we sort of like we interviewed them and asked them for what they need and performed what we call a goal and task analysis to sort of make sure that the system we're building is in line what neuroscientists want to do. So the first goal that we found was to search for particular instances of a motif in a brain network. And when doing that, it's particularly important that in addition to just defining the circuit architecture, we also want to sort of define what sorts of neuron types are involved in that particular motif, where the synaptic connections are happening. So whether a synaptic connection is happening in the optic lobe is very different than if the synaptic connection is happening in the central brain. And that has some sort of physiological meaning. And then finally, it is also extremely critical to be able to set how strongly two neurons are connected, like whether they just have 5 synaptic connections with each other or one has a very different meaning. Okay, so based on this particular goal, we've implemented this sketching interface where a user can draw a network motif, here in this case, a 4-neuron motif, where a user is now connecting these neurons in a particular wiring pattern that they're interested in. And then by clicking on each of the nodes, they can set certain additional constraints like brain region or synaptic strength. Like, for example, neuron A and B need to be connected by more than 20 synaptic connections. And then after hitting the search button, our tool will then go into the connectome and search for instances of that particular pattern. All right, the next goal that we've identified is, I find extremely interesting, and that is how can users correlate the motif connectivity that they've drawn to the actual neuron morphology, what they're seeing in the connectome. And what that means becomes very clear if you look at this example. Like a user has drawn this particular connectivity pattern and then found this instance, these neurons that implement that connectivity pattern. And at least to me, it's very unclear how that particular pattern is implemented or embedded in this set of neurons. And understanding how that works is pretty important. And then to address this, we've designed this focus and context approach in Vimo, where after drawing a particular pattern, a user can inspect this, an instance of that pattern in 3D. And then by dragging this slider here on the bottom, the user can first like prune away all unrelated branches to that particular connectivity pattern. And then by dragging the slider a little further, it would spatially pull those neurons apart and draw links between the pre and postsynaptic sites. Now you can see how it becomes much more visible how this like triangular pattern is like implemented in this set of neurons. Okay, and then the third goal was to trace motif occurrences in synaptic chains. So what that means is, how do particular instances of a pattern spatially relate to each other in the connectome? And for the sake of time, I want to refer you to the paper here if you're interested in that particular one. Okay. Now, we've evaluated Vimo in a case and user study with 7 neuroscience domain experts from HHMI Janelia and the Harvard Center for Brain Science. One of the aspects of this evaluation was an exploratory study. And here's a screen capture of one particular motif that one of the users found that is a recurrent connection in the from visual input neurons that this particular user was interested in. And we've also evaluated certain aspects of the system with our users. One overall sentiment was that people thought that Vimo really improved their understanding of how certain motifs are sort of like embedded in the 3-dimensional structure of neurons. That was an overall positive point. A bit more on the critical side, more experimental features like, like drawing these like links between the pre and postsynaptic side were a bit more debated between the neuroscientists on whether that's useful or not. All right, we found in the user study that a lot of folks were interested in that type of work, and we didn't want to, publish the paper and then, put it in the shelf. So we collaborated with folks from HHMI to integrate Vimo into a larger scale connectome analysis platform called Newprint. And Newprint hosts pretty much all large connectomes and makes them accessible to a wide audience using circuit analysis, interactive exploration. And so now users can query for motifs using Vimo in that analysis platform as well, which was very exciting to see that folks were interested in that line of work. Okay, Now I also want to highlight some limitations of the system, which is that for a very simple connectivity pattern, this is probably the most simple pattern that you can think of, sort of the graph abstraction that VMO uses by mapping each neuron to a single node and then having the synaptic connections as edges. When we look at instances of that pattern, what we often get is extremely high morphological variability. So, for example, in Motif instance number 2 here, these two inputs are spatially very far apart. And on this Motif instance number 2, they may even be entangled or like very close together. And some of the feedback that we got during our user study was that these morphological differences matter when you want to analyze specific types of connectivity motifs. And so that this limitation is now motivating my last contribution that I want to explain briefly now, which is how can we sort of like integrate the spatial or the morphology aspect of neurons into these motif analysis queries. Okay, and I'm going to use this particular example to make my point that the spatial arrangement of neuron and synaptic connection actually really matters. And I'll use this shunting inhibition motif as a case study. And shunting inhibition is a sort of motif in the brain that brains generally use to blend out information that they don't really want to listen to at a particular point in time. So consider this blue neuron. It has a dendrite that receives inputs from other neurons, cell body or a soma, and then an axon that sends out a signal to other neurons. Now, it's important to know that the information flow here is from left to right. So dendrite receives, axon sends signals. And consider this green synaptic connection here by axon 1 that tries to excite the blue neuron. However, in the case of another synaptic connection appearing further downstream, like axon number 2, that makes an inhibitory connection to the blue neuron, so the task of the purple neuron is sort of to shut off or shunt the excitation that's coming from the previous connection. And maybe the key point here to take away is that the sort of order how these two neurons connect to this particular blue neuron is very important in order to implement this circuit motif. So the spatial arrangement here actually matters. And based on that insight, or based on that limitation that we previously had in VMO, we've designed another tool called WOMO that sort of combines connectivity between neurons, but also looks at morphology of neurons. So this is, again, a very simple hello world example. In Momo, we got one blue neuron that makes like a fork. And on each side of the fork, we get one synaptic input connection. And then using Momo, we can search for instances of that pattern, and we get much more morphological consistency in those instances. Now, how do we actually compute these patterns? Or how do we compute motifs over morphology and connectivity at the same time? And we do that by defining this neuron morphology aware graph, because by formulating this problem as a graph, we can then still use all the algorithms that we previously used in VMO, like conventional subgraph isomorphism. Okay, so in this graph represents each neuron segment as a node. And so by doing that, the graph sort of reflects this branching architecture for each particular neuron. And then in addition to mapping the branching structure to the graph, we also say like which segments connect to each other, and those segments are then also connected by edges in our graph. And importantly, what's the key difference to the previous system is that previously neuron A would have been represented by a single node in our graph. Now, using this additional morphology-aware graph, neuron A and neuron B are represented by a sets of nodes that sort of reflect how it's wired. Here's how this looks on a mammalian neuron of the mouse. So each of those segments is colored using the segments are colored differently. And for a small subsection here, I show how this graph is structured. This is how Momo works. It's a Jupyter notebook library because we also found that folks really like to have this like programmatic control over their queries. So that's why we designed Momo as a Jupyter notebook library. And here, a user is drawing a simple morphology-aware motif and then searches for instances of that particular pattern. And then they can inspect instances in 3D, like how are these patterns implemented in the morphology of the neurons. And Momo has this feature where you can like hide away certain structures that are around instances that are found. And so here, by hiding the surrounding segments, this motif structure becomes much more clearly visible. Now, we've also evaluated Momo using with four neuroscience domain experts from these institutions, and we've applied it to motifs such as shunting inhibition and lateral inhibition. Here's a screen capture of how that looked like during the case study, where a user is inspecting a potential case of shunting inhibition, is the motif that I explained earlier. So they've found various instances that they wanted to investigate and then looked at them in 3D and sort of studied whether this particular set of neurons would actually be a shunting inhibition motif or not. Okay, so now I've presented two projects that were tackling the analysis problem. And now briefly summarizing my contributions in that space. So we've conducted a detailed goal and task analysis for motif analysis in connectomes with neuroscience domain experts. We've defined a specific motif analysis workflow that includes a flexible motif searching approach and a focus and context scheme for mapping the circuit architecture to the actual implementations of those circuits. We've proposed a new type of graph-based representation in connectomes that jointly encodes neuron morphology and connectivity. And finally, both tools, Vimo and Momo, are open source available either through the prototypes that we've developed and for some cases also integrated in larger scale systems that are used daily by many people. Okay, so I've talked to you about three of my contributions on the proofreading problem and the analysis problem. And now I want to offer sort of some overall reflections on some of the work that I've been doing. And the first one is that I really think that machine learning in connectomics is much more than image segmentation. So historically, lots of the attention of machine learning in connectomics has gone into image segmentation. But I think that is one particularly important use case, but there is also much more that we can do using these very powerful algorithms, such as thinking about how we can improve the proofreading process or even do some machine learning guided motif discovery or analysis in connectome. My second point is that I think data abstraction here really matters, especially in a scenario like in connectomics where data really is abundant. So one has to think really carefully about sort of which data sources do I want to take into account in order to answer my questions. Like, is looking at the microscopy data actually useful, or does it just make my system much harder to use? Or is it just enough to look at the graph circuitry? And these sort of different data abstraction levels call for very different algorithmic approaches. but also different biological questions may require different data sources. And it's important to think about that beforehand. And then finally, I want to make the point that I think data exploration is not pattern discovery. And what do I mean by that? I think all the analysis tools that I've been proposing are data exploration tools, which is you can sort of like look at a particular hypothesis that you already have in mind and sort of verify whether that holds for a set of particular cases. But in my mind, I think pattern discovery would be a more maybe comprehensive approach where you look at the entire large-scale data set and try to like enumerate or find certain prevalent patterns in the data that you have not known before. And I think those are two different types of approaches of tackling a particular problem. And I would label my tools more as data exploration tools. Okay, talking about future work very briefly. I think there's lots of exciting work. I'm just outlining 4 of those here. I think right now the way we reconstruct connectome data sets is very much through like small specialized machine learning models. I think there's a large opportunity to think about vision foundation models in connectomics, especially because we're at a point right now actually where we have a lot of data. There are cool approaches for multimodal automated proofreading. So I've been working with folks at E11. It's a startup that does connectomics, and they have this super cool approach of sort of like multiplexing certain neurons in a data set. And that can help, or that will certainly help for automating this proofreading process and combined, for example, with these shape-based approaches that I've presented. This is a very exciting avenue of new work. Finally, I think ML-based motif discovery and analysis is an extremely exciting topic, and also using building tools for comparative analysis in connectomics, especially now that we have many different data sets available across sexes, across developmental stages. It becomes very interesting to sort of like compare the circuit architecture and maybe even answer questions on like how individual is a particular connectome to a particular specimen. Okay, and that's it for now. I'm excited to look forward to your questions right now. I will take questions from the audience, and then afterwards I have some acknowledgements that I also want to present.